Hello, we're glad you've joined us for this live webinar, Standardizing Microbiomics, Removing Bias in Collection, Purification, and Analyses. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Zymo Research. Zymo Research Corporation, based in Irvine, California, has been providing the scientific community with comprehensive nucleic acid solutions for DNA and RNA research and epigenetics for more than 2 decades. The company's unique achievements include development of the first available microelution spin column technology for nucleic acid purification. In 2000, Zymo was among the first companies to offer products for epigenetics-based research. In 2011, the company launched its genetic and epigenetic services with state-of-the-art next-gen sequencing and bioinformatics technology. To date, Zymo maintains the most comprehensive offering of services for genetic, epigenetic, and transcriptome analysis. To learn more, visit www.zymoresearch.com. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can pose questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window or report your problem by clicking on the green Q&A button lower left. I now present today's speakers, Ryan Kemp, MS, and Shuiquan Tang, PhD. Ryan Kemp is the Director of Nucleic Acid Solutions for Zymo Research. Kemp joined Zymo in 2012 as a research scientist and has since led the development of multiple novel DNA and RNA purification technologies. His background is in organic chemistry, where he developed artificial receptors for anion recognition. In his current role, he has been instrumental in development of many of Zymo Research's innovative products, including Zymo Pure Plasmid DNA Kits, DNA RNA Shield, and technologies inside many of the quick DNA kits. Kemp also contributed a textbook chapter in sample preparation techniques for soil, plant, and animal samples, published by Springer in 2016, discussing techniques for nucleic acid isolation. Dr. Shuiquan Tang is a research scientist at Zymo Research. He received a PhD in the Department of Chemical Engineering and Applied Chemistry from the University of Toronto. Dr. Tang joined Zymo Research in 2014 and has since been instrumental in the research and development of Zymo's microbiomics and metagenomics programs. His past experiences include work in the fields of environmental microbiology and biological process engineering, with an emphasis on bioinformatics, metagenomics, and anaerobic cultivation. Most recently, Dr. Tang has been involved in the development of Zymo Research's microbiomics portfolio. He has multiple publications in different journals, including Frontiers in Microbiology, Applied and Environmental Microbiology, and Philosophical Transactions of the Royal Society of London, Series B, Biological Science. Ryan Kemp will now begin his presentation.
Good morning, everyone. Thank you for attending Zymo Research's webinar on microbiomics. Today we'll be discussing our efforts basically to remove bias in collection, purification, and analyses. So with the massive boom in microbiomics over the past several years, you can see almost 20,000 citations in 2015 alone, and also major growth in just the number of raw searches in epigenetics versus epigenetics based on Google Trends, almost matching it now in 2016. However, despite this major growth, there's been a lag in the development of standards and standardized methods for enabling reproducibility across studies. And in a study a couple years ago presented by Science News, the author submitted samples to 2 major commercial organizations, American Gut and UBiome. And you'll notice that their samples submitted to American Gut, they found that the firmicutes were about 30%, while at UBiome they observed something like 70%. So with this sample, they're seeing such huge differences, but there's no standards to actually determine which one's actually true. So even from these major commercial organizations, there's no reproducibility, and this has become a major concern in the microbiomics field thus far. So therefore, there's been a real call for standardizing microbiomics in the short term, and FDA has been calling for it. NIST, the National Institute of Standards and Technology, has been working on it. They had a workshop in the last fall. They have another workshop coming in this fall on the standards for microbiome research. The MBQC project, which is the Microbiome Quality Control Project, led by many of the people who are also leading the Human Microbiome project, are also calling for standardization and the requirement for standards to create standard methods. The ABRF MGRG group has also been developing standards and working on standardized methods as well. So as you can see, many groups are actually calling for this so that you can begin these longitudinal studies and have reproducible work between groups. In an assessment of a U.S. microbiome research article that was recently published, one of the major calls was for standardized protocols and high-throughput tools were commonly identified as needs, and along with longitudinal and functional studies and interdisciplinary research. But in order to have these studies, methods must be reproducible between different labs. In an article, baseline study published by the Microbiome Quality Control Project group, some major comments that they had were microbiome research has grown exponentially over the past several years, but studies have been difficult to reproduce across investigations. Addressing the sources of variation in microbiota profiling is critical for optimizing protocols. Unfortunately, variation at each step in the pipeline is enormous, from physical specimen collection and processing to computational quantification of microbial communities. Basically, what they're getting at is the entire workflow, which is a pretty long workflow, is prone to significant errors and biases, and that's what we'll discuss a little bit here, is where some of these errors can occur. So the composition at the time of collection can be changing due to microbial growth or decay. You can have nucleic acid degradation, and then actually where and how a sample is collected can also affect or bias the results or cause erroneous data downstream. So it all starts from the point of collection. During the extraction process, non-uniform lysis of organisms of differential hardiness can bias results, misrepresenting certain organisms or certain communities as being biased towards certain easy-to-lyse organisms versus tough-to-lyse, etc. Library preparation, you can have bias or error due to PCR chimera, primer degeneracy, or biased DNA fragmentation. Of course, sequencing platforms can potentially have bias. And then in the bioinformatics pipeline itself, you can have error in taxonomy assignment or database errors, etc. But then in addition to this, you actually have the potential for reagent and process contamination. So depending on the particular samples you're working with, this can be a much bigger issue, especially if you have low biomass. So you can see there's challenges throughout the entire pipeline that everything needs to be dealt with, and you need good standards to evaluate this. And that's actually what Zymo began a couple years ago when we first started really emphasizing and developing a workflow for microbiomics. We realized that there was not an abundance of commercially available standards, and without good standards and controls, there was no way for us to optimize even our own workflow. So that's actually where we began, was developing our own standards and controls so that we could then begin to optimize the workflow from collection all the way through bioinformatics. And ZymoBIOMICS is really just what we've coined our term for our total solution from collection to analyses. So next I'll talk a little bit about the standard and some specifications about its construction and how we characterize it. So the ZymoBIOMICS standards, we actually have two of them. One will be the microbial standard, which is the cellular standard, and then also we have a DNA standard. There's 10 organisms in each. They're well characterized, and they're predefined composition, meaning we're choosing what goes in, and then we characterize it after the fact to double check, but basically we know what's going in to begin with. If you look to the left here, we have a genomic abundance of 12% for all the bacteria and 2% for both the eukaryotes. There's 3 gram-negative bacteria, 5 gram-positive, and 2 yeast strains. This was chosen very specifically to give us a range of different organisms so that we can look at differential hardiness during the lysis efficiency. There's also a wide range of GC content, so that allows us to look at problems potentially in library preparation, et cetera, due to biases there from 15% to 85%. Another thing to note is we actually chose specific organisms that are notoriously difficult to lyse, like some of the yeast organisms we chose, which are very hardy but also quite large. And then, for instance, Listeria monocytogenes, which is quite small and also quite tough to lyse, notoriously difficult. So this range allowed us to create an exemplary model for looking at a wide range of organisms. Of course, it doesn't test everything in the world, which is impossible, but it creates a decent model moving forward. This also contains 7 pathogens, all of which are inactivated by the DNA-RNA shield, which is present, so it allows for safe transport. So how we construct this, everything's coming from, of course, a single colony. Then we do the pure culture cultivation, and then we quantify. In the case of the microbial standard, we quantify by 2 methods. We actually do an extraction and look at the DNA, and then we also actually do cell counting. We use both of those to determine our microbe pooling. And then after we actually pool, we then characterize again using shotgun metagenomic sequencing. And then for the DNA standard, we do a pure culture cultivation again. We extract the DNA, quantify using qubit, and then pool, and then characterize after the fact as well. We actually found that our DNA standard actually acts as a QC measure for our microbial standard because once we validated our workflow in front of the extraction pipeline and we knew that we had accurate results, we could then use our post-pooling characterization as another measure of validating our microbial standard, which is more complicated to create. This is just looking at the actual composition and our by shotgun metagenomic sequencing and also 16S. And it's on the left you see the theoretical, on the middle you'll see the DNA standard and then the microbial standard. So this is actually the real extraction where we extract it using one of our kits, and you notice that the profiles are matching very, very close to the theoretical. During our early validations, before we had optimized the kit really for microbiomics, we actually were doing a mixture of things where we actually looked at enzymes and chemical lysis and bead bashing all together in the early validations of this to ensure that we had 100% or close to 100% lysis of all the organisms. And on the right you'll see the 16S data, which also closely aligns with the theoretical. So then we decided we were going to look at the impurities within the standard to ensure that the output, whatever people are seeing with using our standard, is in fact an impurity versus say, errors in our construction of the standard. So we did 178 million reads using shotgun sequencing, and we used MOTU profiling. The reason why we chose this was because of the very low false positive rate. And this was a method developed by Sunagawa et al. In 2013. So if you notice here, we had 1 MOTU count for P. acnes, which is extremely low. Therefore, again, this emphasizes the confidence that you could have if you're using these standards that anything you're seeing besides the organisms that we state are present are in fact an error or a bias or something in your system, your workflow, which allows you to optimize and validate your workflow using these standards as we have. Interesting that it is P. acnes because this actually could have occurred even during the process itself because you have to do extractions, library prep, and as you go through that whole process, any one of these places could potentially have a small bit of P. acnes contamination, which is a human skin organism that's commonly found. So next I'll discuss a little bit about sample collection. And DNA RNA shield is our reagent that we really created for all molecular testing, for that matter, for DNA and RNA, but really it has major advantages for microbiomics. And the reason is that it preserves both the DNA and RNA at ambient temperature. It inactivates all the organisms present, preventing growth, any decay, or also prevents any pathogens from being active after the fact, so that way you can ensure safe transport of your samples no matter where you're collecting them. And an example of a use of this was actually during the MERS outbreak in the Oman Desert. The Army actually was ordering a lot of shield from us at the time because they were using it for collecting the nasal swabs from the camels. And two of the major reasons why they needed it was because of the hot desert temperatures. They needed to stabilize nucleic acids, of course, but also they needed to inactivate the pathogen for transportation. So DNA RNA Shield takes an unbiased molecular snapshot at the time of collection, meaning it really freezes the sample at the time of collection. This allows you to get that picture immediately as soon as you collect it. There's no changes, and I'll show a little data about how fast the sample in fact can change in the case of a fecal sample. If you're not collecting it in a media that immediately stabilizes nucleic acid and activates all the organisms. Works in blood, urine, works with stool, soil, swabs, essentially any sample you can imagine it's functional with. So to kind of reiterate, it preserves nucleic acids at ambient temperature, inactivates the pathogens, all the organisms in fact, and streamlines purification. And I want to emphasize streamlined purification because of the article that I pointed out earlier where they noted that high throughput methods were a commonly identified need for microbiomics. Well, if your reagent requires any reagent removal, then it's very difficult to streamline the purification due to the fact that you have to introduce either centrifugation or something else to remove reagents. In addition to that, anytime you're causing phase separations, it introduces bias, and there's a number of articles written on this already. So DNA RNA Shield is highly automatable. There's no reagent removal. You can go directly into your purification method. So some data that support the DNA RNA Shield's ability to stabilize nucleic acids at ambient temperature. So we have some saliva data here, RT-qPCR and qPCR for both saliva, stool, and tissue. This tissue is actually solid tissue chunks here, indicating the ability of Shield to actually penetrate a solid or semi-solid sample. And then on the bottom right, you'll see some data exemplifying how Shield not only is stabilizing via RT-qPCR, but we can actually look at it visually on a gel at 37 degrees C. Within 3 days, you already have a significant degradation of the RNA, as indicated by the disappearance of the ribosomal bands and smearing. So how this applies also specifically to microbiomics is if you collect on the top here you have a stool sample collected in DNA RNA Shield, and we look at it for a month, and you see the profile doesn't change at all. The community profile remains the same. However, in the case of a stool sample collected not in Shield, just left alone, you see even within 1 day, the proteobacteria and the actinobacteria already start changing their relative composition quite quickly, causing pretty significant bias in these results towards organisms that would normally be found in much, much lower abundances. And again, pathogen activation or organism inactivation as a whole, but we've tested various bugs, influenza, Ebola, HSV, some BSL-2 gram-positive, gram-negative organisms, Shuiquan-producing E. coli, Listeria monocytogenes, Cryptococcus neoformans in the case of yeast we've tested. And you notice the DNA RNA Shield enables complete inactivation of these organisms for safe transport. And actually in the lab, also safer for the lab techs or researchers actually working with the samples. So discuss a little bit about purification now. So Zymo really spent significant effort in validating our purification line of products for microbiomics, because previously microbiomics wasn't the major use of, say, a fecal or a soil kit. People were doing many other things, but microbiomics as a whole has grown so much, the needs have actually changed. And so Zymo really sought to meet these needs by developing a kit aimed at the microbiomics community specifically. So we aimed at non-biased lysis and efficient purification and low bioburden, so that for low sample inputs, so you have very low contamination so that the reagents are all clean. And in addition to that, also efficient inhibitor removal, so that two of the most common sample types, which are fecal and soil samples, are rich in inhibitors, inhibiting PCR, et cetera, which are important for those methods. So a little background, if you look at chemical lysis on the right here compared to our theoretical, this is actually using our standard, you notice that chemical lysis is inefficient at lysing the gram-positive organisms. You have a huge overrepresentation of the gram-negative organisms. While the ZymoBIOMICS DNA mini kit that we are releasing will use mechanical lysis. However, mechanical lysis is not all created equal. That was actually a lot of our validations, was creating the correct system, the buffers, the beads, etc., to provide an unbiased, efficient lysis of all the organisms. We actually found that even not all bead beating devices are created equal, and we actually recommend that use the standards to even titrate your bead beating device, how long and what speed you should be using your bead beating device to optimize your yield, but more importantly to ensure you have an unbiased representation of what the sample actually is. High-speed units will require less time, of course, and lower-speed units require much more time. So then we also looked at 3 of the most common methods used in the literature, basically the 3 most cited methods for fecal and soil samples. And the one on the right is supplier Q. That's actually a chemical lysis method. Even though it's quite commonly used in this field, we were surprised by it, but that's what the literature is stating. And due to the fact that it's a chemical lysis method, you actually see bias, again, towards these gram-negative organisms, as you'd expect. Supplier M is actually using mechanical lysis, and this is the most cited method and for the microbiomics specifically. And you notice it also is biased towards the gram-negative organisms, inefficiently lysing many of the gram-positives. And then the HMP protocol, which is the Human Microbiome Project protocol, is actually using the supplier M kit here, but also adds a heating step. And what we found, or what we suspect is that the heating step actually further lyses many of these gram-negatives, but isn't in fact actually further enhancing the lysis of many of the gram-positives. Again, what we'd like to reiterate is that many of these things weren't actually developed, these original kits weren't developed for this specific use of microbiomics. So that's why Zymo had to develop a standard in order to validate the methods to ensure that we had a method that was unbiased and was really looking to provide a tool that caters to this community specifically as the needs have emerged. Then we decided to look at the same profile using the four same methods, looking at an actual fecal sample. And you can notice the ZymoBIOMICS DNA mini kit, which was unbiased using our standard in the previous slide. We actually have high yields, but also a much higher abundance of firmicutes. So first I'll discuss the yield a little bit. This yield is directly indicative of the fact that the lysis efficiency is greatly enhanced. And again, this was a key point because without efficient lysis of all the organisms, or a vast majority, you risk bias of your sample. And then in the case of the fecal sample, you notice that this data closely aligns or agrees with the data we observed with the standards, with the firmicutes, the gram-positives being lysed more efficiently with the ZymoBIOMICS DNA mini kit, and the other gram-positive, the actinobacteria. And you notice that the bacteroides actually represent a very small fraction of the sample. However, the other methods show bacteroides actually composing a much larger fraction and misrepresenting what is actually present. In addition to that, we've upgraded our lysis system as a whole to ensure that the DNA is actually of higher integrity. Of course, the bead beating, the mechanical homogenization is actually still going to cause some shearing, but through some optimization, we improved the average size of the DNA coming out of the sample, with a majority of it appearing 10 kb and above, making this potentially amenable to some of the third-gen sequencing methods. Then looking at also our PCR inhibitor removal ability. So common PCR inhibitors are polyphenols such as humic acids, fulvic acids, tannins, and all these things are very common in fecal and soil samples. And this is one of the richer inhibitor samples that we've dealt with, and that's why we use this here as one of our controls, actually to optimize our whole system. And what this is we're looking at basically a standardized input of everything using a plasmid. So all the cycle numbers should actually be identical, and then we increase the amount of LU weight present, and as the LU weight increases, you would either expect if there's many inhibitors present, you'd expect to see the inhibition increase or no amplification. With the ZymoBIOMICS kit, you see that the sample remains at 17.5 cycles approximately, and supplier M and P, they're still good at about 10% of the LU weight. However, as you increase the amount of LU weight, you notice that the samples become completely inhibited. Lastly, we've also we worked on developing a polymerase that was low bioburden. This particular polymerase has less than 2 femtograms of DNA, which is less than a single bacteria coli genomic equivalent. which makes it ideal for many of the microbiomics work. And that concludes my section, and then I'll pass it off to Shuiquan now.
Thank you, Ryan, for that informative presentation. I will now turn it over to Dr. Tang for his presentation.
Good morning, everyone. So this is Shuiquan Tang from Zymo Research. I will continue the presentation after Ryan. So my part of presentation will focus on Zymo's effort in Zymo's effort in standardizing library preparation and analysis in microbiomic analysis. So generally, there are two ways to profile a mixed microbial community. One is based on targeted sequencing, and the target region can include 16S rRNA genes, fungal ITS regions, and eukaryote 18S rRNA genes. The other approach is based on shotgun sequencing. Both ways have advantages and disadvantages. The targeted approach is robust and of low cost, but it also has limited coverage and limited resolution. For example, the 16S rRNA gene targeted sequencing cannot detect eukaryotes and virus. And in most cases, it can only identify bacteria to the genus level. On the other hand, shotgun metagenomic sequencing sequences all available DNA. And because of that, in theory, it can identify all organisms except RNA virus, and it can trace microbes to the strain level. However, it also has some disadvantages. For example, it has problems of incomplete database and host DNA contamination. It also has a relatively high cost. At Zymo Research, we offer both as sequencing services. All our workflows and pipelines have been optimized and validated using the ZymoBIOMIC Microbial Community Standard. In the following, I will introduce some examples of our optimization efforts for these workflows. So normally, 16S rRNA gene targeted sequencing or 16S profiling is the first choice for many researchers looking for profiling a mixed microbial community. The library preparation process of 16S profiling is basically a PCR amplification process. As you know, in a PCR reaction, the amount of PCR product mainly depends on two things: the amount of initial templates and the number of PCR cycles. The amount of initial templates in an unknown sample can vary from a few copies to millions of copies. For convenience, and in order to have enough PCR product from a variety of samples, many researchers choose to have a fixed and relatively high number of PCR cycles for all different samples, especially in the format of 96-well plate. For example, in the Human Microbiome Project protocol, it says like 30 cycles. And in the Earth Microbiome Project, they recommend the use of like 35 cycles. Running the PCR reaction for 35 cycles can allow you to have enough PCR product for sequencing even from a highly diluted sample. But this will overrun the PCR reaction for those samples that are not as diluted. And overrunning the PCR reaction can cause the formation of PCR chimeric sequences. The figure on this slide shows how PCR chimeric sequences can grow if a PCR reaction runs away. The PCR reactions shown in this figure have about 10 nanogram of genomic DNA input from the ZymoBIOMICS standard. And because of the use of standard, we can identify all PCR chimeric sequences using U-time and the reference rRNA gene sequences that were present in the standard. And as you can see in this figure, running the PCR reaction for just 30 cycles can cause the PCR chimeric sequences to take up more than 35% of the final PCR products. Many researchers choose to rely on bioinformatic tools such as U-time to filter out these chimeric sequences. However, it's highly questionable how effective that approach can work for all unknown samples. At Zymo Research, we don't use a fixed PCR. We don't use a fixed number of PCR cycles for all different samples. We adjust the PCR cycles for each individual sample. We develop a process that allows you to adjust to control each PCR reaction in a 96-well format, and therefore controlling PCR chimera formation from its source rather than dealing with the problem afterwards. In 16S sequencing, normally the more you sequence, the more taxa you will be able to identify. However, many of those taxa might be derived from artifacts in 16S sequencing. The potential source of artifacts in 16S sequencing can include PCR chimera, reagent and process contamination, sample cross-contamination, barcode contamination, MySeq run-over-run carryover, PCR errors, and sequencing errors, etc. The defined composition and the extremely low impurity level of the ZymoBIOMIC standard allow us to assess the artifacts in 16S sequencing thoroughly. This pie chart shows the microbial composition of the ZymoBIOMIC standard when sequenced and analyzed using a regular workflow in which no measures were in place to control these artifacts. And as a result, as many as 43 bacterial genera are identified, while the standard should only have 8 bacterial genera. This pie chart on the right-hand side shows the microbial composition of the ZymoBIOMIC standard when sequenced and analyzed using our current workflow. And in this case, measures were in place to control low bioburden, to limit PCR chimera formation, to have a very good MySeq run, and also to have other bioinformatic controls. And as a result, we only identify 8 bacterial genera, which agrees perfectly with the composition of the standard. So one of our collaborators is a big core facility which routinely process a large amount of samples for 16S sequencing. And they participated in a beta test of our ZymoBIOMIC standard. And the initial profile they found in our standard looked like just the pie chart on the left-hand side. And obviously, they have some optimization work to do for their own workflow. Now let's talk about shotgun metagenomic sequencing. The library preparation process of shotgun metagenomic sequencing is a much more complicated process. It contains at least 5 steps: DNA fragmentation, end repair, A-tailing, adapter ligation, and PCR amplification. The library preparation kit, called Nextera XT was provided by Illumina for shotgun metagenomic sequencing. And this kit is very popular because it shortened the whole process from 5 steps to 2 steps, which is segmentation and PCR amplification. However, convenience might come with a price that many researchers do not know. The sequencing coverage bias can occur because of GC content variation. We realized there might be some problem with the Nextera XT kit when we compare it side by side with our internal method for shotgun library preparation when using the ZymoBIOMIC standard for shotgun sequencing. The microbial composition shown on this figure was determined by mapping the raw reads against the reference database. They were contained in the standards. And while the results from our internal library preparation method agrees almost perfectly with the theoretical value, the results from the Nextera XT kit has some disagreement. And the biggest difference comes from the organism of Staphylococcus aureus. And this organism happens to be the organism that has lowest GC content in our standard, which only has about 32.7%. By mapping the raw sequencing reads against the reference genome, one can visualize GC content variation and sequencing coverage side by side. And this figure focuses on a region, a 10 kb region from the genome of Staphylococcus aureus. The panel on the top shows the GC content variation, and the bottom 2 panel shows the sequencing coverage variation. Now, The panel in the middle shows the case of next sterile XT kit, and as you can see, the sequencing coverage increases when GC content increases and decreases when GC content decreases, so on and so forth. And they basically share a very similar trend. But you don't see this phenomena in the case of our internal method. As we mentioned earlier, such GC bias can cause a bias in the estimation of a microbial abundance. And in the case of Staphylococcus aureus, for some regions that have extremely low GC content, the researchers might not be able to recover any sequence from those regions. And this is definitely problematic in some applications. The ZymoBIOMIC microbial standard contains multiple organisms that span a wide GC content range, from as low as 15% to as high as 85%. And this allows us to assess GC bias in a complete way. So the figures in this slide compares the two library preparation process regarding GC bias. Over 3 genomes were contained in a microbial standard. These 3 genomes were picked to represent GC content low, medium, and high. The pie charts on these figures represent the GC distribution in each genome, and the curves represent the normalized sequencing coverage distribution along GC content. Ideally, if the sequencing coverage has no bias caused by GC content, the normalized GC content should always equal to 1. And we can see this is almost the case in the case of using our internal method. However, in the case of the next sterile XT, we've seen under representation for regions of both low GC content and high GC content. So within our ZymoBIOMIC sequencing services, we also provide a state-of-art and comprehensive bioinformatic and statistic analysis. This slide gives some examples of typical outputs. Here's a summary of our 16S RNA gene targeting sequencing service. Because of the use of the ZymoBIOMIC standard, we guarantee the data of highest quality and strict controls to maintain low bioburden allow us to deal with samples that have extremely low biomass. And our service is a total solution. The customer just needs to send out the samples and we can take out the rest. With all these great features, we also provide a good price. For our services, we recommend our customer to connect samples in DNA analysis shield for unbiased microbial preservation and pathogen inactivation. We use ZymoBIOMIC DNA minikit for unbiased DNA extraction, and we use ZymoBIOMIC low bioburden DNA polymerase for low bioburden control, and we limit PCR cycles to control PCR chimera formation. The DNA extraction and library preparation were performed in ultra-clean room to prevent process contamination. And most importantly, all our workflows and pipelines have been optimized and validated using the ZymoBIOMIC microbial standard. This is a summary of our shotgun metagenomic sequencing services. So basically, we use an internal method for unbiased library preparation. We use HiSeq for high throughput sequencing and sophisticated bioinformatics tools for data analysis. So in the end, I would like to finish my presentation with a real story from Boo Boo. So Boo Boo is a pet that belongs to my colleague Mark. So one day, Boo Boo was diagnosed to have the so-called atopic dermatitis on one of his paws. Basically, it's a skin disease assumed to be caused by allergic reaction. And Boo Boo was given like anti-inflammation drugs for the treatment. My colleague Mark was suspicious about the diagnosis and decided to conduct a simple microbiomic study. So he connects samples using swab from the disease port and save the samples in DNA and RNA shield. Boo-boos recover slowly for about 1 month, and after recovery, Mark connects the sample from the same area and then send those samples to us for microbiomic sequencing. We sequence the sample with both CKNS RNA gene targeted sequencing and fungal ITS targeted sequencing. While the fungal community has no obvious difference between the disease sample and the control sample, there is a dramatic difference between them in the bacterial community. So basically, the bacterial community in the disease sample was dominated by 2 bacteria genera, Streptococcus and Staphylococcus. Many members from these 2 bacterial genera are common pathogens, and they're also commonly found in the case of like co-infection. So members from these 2 bacterial genera are all gram-positive organisms. They are very tough to lyse. And I doubt I would question like if one can see such a dramatic difference between these samples if not using like if not preserving the samples in DNA shield and process them using the ZymoBIOMIC ZymoBIOMIC DNA mini kit. So hopefully in this presentation, we have convinced you the importance of using the reference material in microbiomic studies. And using the ZymoBIOMIC Microbiome Standards, Zymo Research have created a complete and unbiased solution for microbiomic composition profiling. So we have DNA shield for sample connection. We have ZymoBIOMIC DNA mini for DNA extraction. We have ZymoBIOMIC low bioburden DNA polymerase for library preparation. We have ZymoBIOMIC Microbiome Standard as a quality control for the whole workflow. And we also offer ZymoBIOMIC sequencing services as total solutions for microbiomic studies. So in the end, I would like to thank all my colleagues at Zymo Research who have contributed to the work presented in this presentation, especially to Dr. Larry Jia, who is our R&D director, and Dr. Stan the Foreman, who designed DNA RNA shield. Next, we would take questions.
Thank you both for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Ryan Kemp and Dr. Shuiquan Tang will answer as many questions as time permits. And the first question is, how do we address microbiomics biases in tissues with low abundances of microbes?
Sorry, can you iterate the question again?
Absolutely. How do we address microbiomics biases in tissues with low abundances of microbes?
So in the case of low abundance, it's very critical to maintain it's very critical to have low bioburden control for all your process. You need to control. You need to have procedure to maintain low bioburden in your reagent and also during the process. As I mentioned in our ZymoBIOMIC sequencing services, all our reagents were manufactured by ourselves, which we have full control, and all the process for DNA extraction and library preparation, we have to do it inside ultra clean room to prevent process contamination.
Is microbial standard a mix of the bacteria and fungi, which can then undergo DNA extraction?
Correct. So we have 2 standards. Oh, I was going to say we have two standards. One is they're mirrors of each other, but one is the extracted DNA, and the other one is the microbes pooled.
Do bacteria keep intact membranes in DNA RNA shield? We are using PMA to remove extracellular DNA.
That's a tricky question. They do maintain an intact cell wall, especially in the case of many of the tougher gram-positives and yeast. But DNA RNA shield is lytic, so that's why it's safe for transport, et cetera. But it's still effective for the evaluation of biases associated with extraction because the cell walls remain intact, of the tougher organisms especially.
How do chimeric sequences form during 16S library preparation?
For this question, so there are several publications investigating PCR chimera formation in 16S library preparation. So PCR chimera is a problem in PCR reactions in which there are a variety of different but also highly similar templates coexisting. One potential mechanism of how they form is that in a step of PCR elongation, early elongation termination results in partially amplified sequences in one cycle, and this partially amplified sequences are used in subsequent cycles as primers for further elongation. When they are leading to a different template, a hybrid product from two different templates is produced. Again, these chimeric sequences are totally artificial, and they dramatically increase the alpha diversity of the sample, just like what we see in the case of when we profiling using our standards. And they will bring they will bring a lot of noise to the data, which might bury important discoveries. They need to be removed and taken care of.
Are ZymoBIOMICS standards optimized for high, about 5%, and low fat, 0 to 1% food samples?
So to reiterate, I think this refers to the extraction technology. And yes, we have appendices and methods for dealing with essentially any sample type, and then we can work with you on that.
How does GC content variation cause bias in sequencing coverage in shotgun sequencing?
So currently, there is no perfect explanation to how GC bias occurred. Some reports have shown that it can come from the process of PCR amplification. Some indicate that it might come from the sequencing platform itself. But according to our results, because we don't see obvious GC bias in our internal library preparation process, we think that GC bias caused by the sequencing platform should be subtle if existing. The next TerraXT kit only has two steps, enzymatic segmentation and PCR amplification, and the bias we observe can come from either or both.
Can I still get enrolled in the beta test of ZymoBIOMICS Microbial Standards, or when will the products be released?
Yes, you can still get involved in the beta test, and the product will be released at the beginning of July. But free samples for the beta test are available now, and you can sign up. You'll notice that we have the ZymoBIOMICS webpage listing available on the slide right now. You can go there for more information.
Is it possible to purchase the microbial and DNA standards for local validation and development work?
We can send you stuff now free of charge for your validation and testing.
Can you tell about a brief principle of RNA-DNA shield?
It's a lytic reagent, so that's the general principle behind why it inactivates things. However, in terms of stability, that's kind of our secret sauce, but it stabilizes both DNA and RNA at ambient temperature for greater than a month.
Does the DNA-RNA shield protect both DNA and RNA of bacteria and fungi in a single solution?
Correct.
And let's just see if we have more questions coming in, or if we will be ending. Give us a moment here. Okay, looks like just a moment. How can you assess whether these methods are, in quotes, unbiased? The gold standard has been highly debated.
Unbiased is used a little loosely here. It's only unbiased relative to our standard. But our standard is including many organisms of differential hardiness, etc. It's the closest that we can get to unbiased without including, say, another 100 organisms or 1,000 organisms. So that's our philosophy on it, is that we're moving towards as close to unbiased as we possibly can, but you're absolutely correct. There's no guarantee it's absolutely unbiased in all contexts 100% of the time. But everything's
long-term
standard. Oh, sorry. And everything's based upon the standard, so that's our criteria.
Have you done long-term studies of the protective effect of the DNA-RNA shield?
Can I get clarification if that is with respect to the stability of the DNA and RNA? And if that's the case, then yes, we have long-term studies. Actually, DNA is stable out for years. RNA is, we guarantee at least 1 month, but actually is stable even longer. And then, of course, we say ambient temperature at 30 days, but there's some variability there depending on if your ambient temperature, say, is 37 versus your ambient temperature is sitting at 15 all the time.
Well, I would like to once again thank Ryan Kemp and Dr. Shuiquan for their presentations. Do you have any final comments?
I just want to thank everybody for attending today, and we hope to see you guys at maybe ASM, and we're also going to be at the NIST workshop in the fall.
Dr. Tang? Yeah, so we are going to ASM next week, so hopefully we can, if you are interested in our topic, we can have more close discussion about more details about our process.
And there was a question about whether or not the slides will be made available, and yes, the slides will be made available. My understanding is also the presentation will be made available.
I'd like to thank the audience for joining us today and for their interesting questions. We would also like to thank our sponsor, Zymo Research Corporation, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through December 9th, 2016. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.